The world watched with apprehension as news broke of HKU5-CoV-2, a novel coronavirus discovered in bats at the Wuhan Institute of Virology. This discovery came just a few years after the devastating COVID-19 pandemic. The scientific community sprang into action, determined to understand this new virus. The discovery underscored the importance of ongoing surveillance of animal populations, particularly bats. The Wuhan Institute of Virology played a crucial role in identifying and isolating this new pathogen. Their work is critical for early detection and pandemic preparedness. As we encroach into animal habitats and global travel connects us, the risk of zoonotic spillover events increases. The world held its breath, waiting to see what this new virus held in store. HKU5-CoV-2, like its infamous cousin SARS-CoV-2, belongs to the beta coronavirus family. These viruses are masters of disguise, their RNA genomes constantly mutating, allowing them to jump from animals to humans.
Understanding the structure of HKU5-CoV-2 is key to understanding its potential for spillover and its ability to cause disease. The virus particle is a tiny sphere, its surface studded with spike proteins. These proteins are the key to the virus's infectivity. They act like keys, searching for the right lock on the surface of human cells. Once they find their match, the virus can latch on and gain entry, hijacking the cell's machinery to replicate itself.
Scientists meticulously mapped the structure of these spike proteins, looking for clues about their binding affinity to human cells. They compared the genetic sequence of HKU5-CoV-2 to other coronaviruses, searching for similarities and differences that might explain its behavior.
Unraveling the secrets of HKU5-CoV-2's structure is a race against time. The more we understand about how the virus is built, the better equipped we are to develop effective vaccines and antiviral treatments. This knowledge could be the difference between a contained outbreak and a global pandemic.
In the realm of coronaviruses, HKU5-CoV-2 bears a striking resemblance to another notorious pathogen, MERS-CoV, the virus responsible for Middle East Respiratory Syndrome, MERS. This similarity, while concerning, also offers a glimmer of hope.
By studying what we already know about MERS-CoV, scientists can gain valuable insights into the potential behavior of HKU5-CoV-2. MERS, first identified in Saudi Arabia in 2012, caused severe respiratory illness with a high mortality rate. Like HKU5-CoV-2, MERS-CoV originated in bats and spread to humans through an intermediary animal, in this case, camels.
The similarities in their origins and transmission patterns raise concerns about the potential for HKU5-CoV-2 to follow a similar trajectory. However, the link to MERS also offers a silver lining.
Scientists have made significant strides in understanding MERS-CoV since its emergence, developing diagnostic tests, antiviral treatments, and even a vaccine. This knowledge base could prove invaluable in accelerating the development of countermeasures against HKU5-CoV-2.
The similarities between HKU5-CoV-2 and MERS-CoV serve as a stark reminder of the interconnectedness of animal and human health. As we continue to encroach on animal habitats, we increase the risk of encountering novel viruses with the potential to spill over into human populations. Understanding these connections is crucial to preventing future pandemics.
Section 4, the ACE2 connection unlocking human cells. The ability of a virus to infect human cells hinges on its ability to bind to specific receptors on the cell surface. In the case of HKU5-CoV-2, as with SARS-CoV-2, the key target is the ACE2 receptor. This receptor, found on cells in the lungs, heart, and other organs, acts as a gateway for the virus to enter and infect human cells.
The spike protein of HKU5-CoV-2, like a key fitting into a lock, binds to the ACE2 receptor with remarkable affinity. This binding triggers a cascade of events that allows the virus to fuse with the cell membrane and release its genetic material into the cell. Once inside, the virus hijacks the cell's machinery, forcing it to churn out copies of itself.
As scientists are meticulously studying the interaction between the HKU5-CoV-2 spike protein and the ACE2 receptor, they use sophisticated imaging techniques to visualize the binding process at the molecular level, mapping the precise points of contact between the virus and the cell. This detailed understanding is crucial for developing drugs and vaccines that can block the virus's entry into human cells.
The ACE2 receptor, once an obscure protein, has become a focal point in the fight against coronaviruses. Understanding its role in viral infection is paramount to developing effective strategies to prevent and treat these emerging threats. The knowledge gained from studying HKU5-CoV-2's interaction with ACE2 could have far-reaching implications for combating future coronaviruses. Section 5, SARS-CoV-2 versus HKU5-CoV-2, a tale of two viruses. The discovery of HKU5-CoV-2 inevitably draws comparisons to SARS-CoV-2, the virus responsible for the COVID-19 pandemic. Both viruses share several similarities, including their origin in bats, their ability to bind to the ACE2 receptor, and their potential to cause respiratory illness in humans. However, crucial differences between the two viruses could determine the severity of their impact on human health. One key difference lies in the virus's transmissibility. SARS-CoV-2 proved to be highly contagious, spreading rapidly through respiratory droplets and causing widespread outbreaks. The transmissibility of HKU5-CoV-2 remains unknown, and scientists are working diligently to assess its potential for human-to-human transmission. Another crucial factor is the severity of the disease caused by each virus. While SARS-CoV-2 led to a wide range of symptoms, from mild to severe and even death, the pathogenicity of HKU5-CoV-2 in humans is still under investigation. Early studies suggest that HKU5-CoV, the 2nd of May, caused milder symptoms than SARS-CoV-2, but more research is needed to confirm these findings. The comparison between SARS-CoV-2 and HKU5-CoV-2 highlights the importance of not jumping to conclusions. While similarities exist, the devil is in the details. Understanding the nuances of each virus's behavior is crucial to mounting an effective public health response. Section 6. Assessing the threat. Expert opinions on HKU5-CoV-2. The emergence of HKU5-CoV-2 has sent ripples of concern through the scientific community. Experts worldwide are closely monitoring the situation, analyzing data, and sharing their insights to assess the potential threat this new virus poses to human health. While much remains unknown, their cautious optimism provides a glimmer of hope amidst the uncertainty.
Dr. Linfa Wang, a world-renowned expert on bat viruses at Duke US Medical School in Singapore, cautions against alarmist pronouncements. It's important to remember that not all bat coronaviruses pose a significant threat to humans, he explains. While HKU5-CoV-2 shares some similarities with SARS-CoV-2, it's too early to say whether it has the same pandemic potential.
Dr. Emmy DeWitt, a virologist at the National Institute of Allergy and Infectious Diseases, echoes this sentiment. We need more data to fully understand the risks associated with HKU5-CoV-2, she says. We need to study its transmissibility, its ability to cause disease in animal models, and its potential to evade the immune system.
The cautious optimism of these experts underscores the importance of a measured and evidence-based approach to emerging infectious diseases. While vigilance is crucial, so too is avoiding unnecessary panic. The scientific community is working tirelessly to gather the information needed to make informed decisions about how best to address the potential threat of HKU5-CoV-2. Section 7. Market jitters, the economic ripple effects. News of the HKU5-CoV-2 discovery sent shockwaves through global markets still recovering from the economic fallout of the COVID-19 pandemic. The memories of lockdowns, supply chain disruptions, and financial turmoil were still fresh in the minds of investors, leading to a predictable, albeit muted, reaction.
Stock markets, particularly those in the travel, tourism, and hospitality sectors, experienced a dip as investors reacted to the news. The uncertainty surrounding the virus's potential impact on global health and the possibility of renewed travel restrictions spooked markets, triggering a wave of selling.
Pharmaceutical companies, on the other hand, saw their stocks surge as investors anticipated a renewed focus on vaccine development and antiviral treatments. The race to develop countermeasures against HKU5-CoV-2 was on, and investors were eager to capitalize on the potential windfall.
The market's reaction to the HKU5-CoV-2 discovery highlights the interconnectedness of global health and the global economy. The COVID-19 pandemic served as a stark reminder that infectious diseases can have far-reaching economic consequences. The emergence of HKU5-CoV-2 underscores the need for continued investment in pandemic preparedness and the development of resilient healthcare systems.